start. Okay, welcome everybody to this third day of the H3Africa Consortium meeting. We've had some amazing talks yesterday and we look forward to some great talks today. So we're starting off the session with one of our previous family members, so Catherine Littler, who was previously at the Wellcome Trust, and she's been part of the H3Africa family for many years, and we miss having her around. But Catherine is now the co-lead of the Global Health Ethics and Governance Unit at the World Health Organization. And today she's going to be talking to us about genomics challenges and opportunities for ethical implementation. Thanks, Catherine.
Good afternoon and a warm welcome. I'd like to first thank you all for the opportunity to speak today. The H3Africa initiative is one of the seminal learnings of my career, and I don't just mean that on a technical level. I mean what I learned in terms of collaboration and value. So I owe the initiative a debt of gratitude. So in some ways it feels like I've come full circle to come back and talk to you all about some work that I led at WHO that really has been influenced, I think, and builds on many of the foundations of the work that has taken place at H3Africa. So thank you again. This is going to be a bit of a whistle stop of the work because there's a lot to go through. I'm going to talk about two things. One is, as I said, some work that's built on the foundations of H3Africa. That is the work of the Human Genome Editing Committee, which was recently launched in summer this year after a 2-year period. So I'm going to talk about the two main outputs and reference a couple of those. One is the governance framework and the other is recommendations. Given how big these are and how little time I've got, I'm going to try highlight some of the key areas and points for consideration that are really relevant to the work of H3Africa and the consortium going forward. So that will be the first and the main bit of my presentation. And then secondly, I'm going to focus on WHO and genomics and really the new opportunities and the opportunities for WHO and H3Africa to collaborate going forward. And I think that really is something exciting. So I'll move to start talking about the work of the Human Genome Editing Committee. And for those of you who aren't familiar with it, it was established by the Director General of WHO in December 2018 and had its first meeting in March 2019. You'll see from the list I put up of the committee member, it is a very broad global committee, multidisciplinary committee, and it was tasked to examine the global standards for governance and oversight of human genome editing from a scientific, ethical, social, and legal perspective, and looking at very broadly, so not just germline inheritable, but also somatic technologies. The committee's drawn from all the WHO regions. As you can appreciate, like much of your work, a lot of the committee members got caught up in the COVID-19 pandemic. So some of the work was slightly delayed, but we are very pleased that it's now been launched and I get the opportunity to talk to you about it today. I'd also like to note that you will see that one of the alumni, one of your colleagues, Associate Professor Jantine de Vries from the University of Cape Town was one of our committee members. So the work of, during its work, the committee consulted very widely with international organizations, regional organizations, ministries, regulatory authorities, civil society groups. We had committee meetings, 3 of which were face-to-face, and then clearly they needed to move to online. We had many webinars with different types of experts who I've started to name. We had consultations on the establishment of the Human Genome Editing Registry, which I will briefly talk about. We had 2 satellite meetings that I think significantly contributed to the findings. One was actually held in Cape Town in late February. That was our last face-to-face meeting, and that was with the sickle cell community. And we do owe them a debt of gratitude, because I think a lot of that discussion flows primarily through the governance framework, but has also fed into the recommendations document. A lot of discussions about access to treatments, really good discussions about inclusion of participants' perspectives that I think was eye-opening for committee members. Issues around stigmatization, ethics dumping were raised, and really the scalability and affordability of these types of technologies as we move forward. We also consulted online about the governance framework. I want to say that, as I said at the beginning, the remit was quite broad in sense of it wasn't just germline and heritable, but the committee felt very strongly, as the DG did, about including somatic. And that's really because we understand there is some very good somatic oversight in certain countries, but there's a real patchwork, and this is where a lot of the health improvements will be in this area. So the committee felt very strongly about including somatic. I think also the DG emphasised looking at the ethical and societal issues associated with governance and really trying to embed them in policy and decision-making going forward. I think it's important to say that the committee's remit did not include a review of matters to do with safety and efficacy of human genome editing. On that, we're very privileged to build on the work of the International Committee on the Clinical Use of Human Genome Editing that was convened by the US National Academy of Medicine, the US National Academy of Sciences, the UK Royal Society, and a number of academies from around the world. And we really built on their work, so we didn't focus on a lot of what was covered by them. And I know that Michelle Ramsey, who's also part of the H3Africa initiative, was a key member of that commission as well. So we do owe we did build on the foundation of their work and many other groups as well. We also tried to make the outputs of the committee not just about now, but looking really at the longer term future use. And it's very difficult, as many of you know, to predict where the science is going to take us, but we felt that was an important component. So the committee outputs, well, the first one really was the establishment of a pilot for a clinical trials registry on human genome editing. And this one's critically important, I think, in terms of transparency. And is one that will need to be built on, because one of the second findings was that a basic research registry would also be very helpful, and this hasn't been established. And I think this is something that the genomics community could also build on, and further discussions could happen on that. That there was a statement by the Director General that it would be irresponsible to use genetically altered embryos for reproduction following the mooting of this, which got wide uptake. And as I said, I'm going to focus on the governance framework and the recommendations document. But there are also 6 reports of the committee's work, and we commissioned reports both on the issue of whistleblowing, but also looking at some of the key science achievements to inform this. So I'm going to move to the governance framework, which is one of the main 2 main outputs of the committee. And the committee worked for 2 years on this. And it worked really to produce a framework that would offer guidance in how to develop policy and tools to implement policy choices. Much of the final product was informed by both our meetings and the 2 online consultations, as I said earlier. The committee felt strongly, it's a very global committee, it had a global mandate, that this framework needed to be scalable, sustainable, fit for purpose at the international, regional, national and local levels, and work in very different settings, but particularly in settings where regulation may be weaker and or where genome editing is not necessarily being pursued at the moment. So this sort of future proofing or thinking about regulation or oversight in advance, I think is critically important. But above all, the framework needed to be practical and usable and really focus on implementation. And I'll come back to that because I think this is an area in which we can work with the H3Africa community going forward. In the discussions, a few points came out that I think are very pertinent to the work of H3Africa, and that is that the data on human genetic diversity and the role gene variants play in health and disease is under different genomic and external environments are foundational to human genome editing. And that these collections are currently not representative of the global population, and really to make these technologies accessible to as many populations as possible, we need to change that. And this comes down to a lot of the equity issues that I think we're all grappling with in research, and was one of the foundational values of establishing H3Africa. So that really came through as a key concern for the development of inclusive genome editing innovations, and something that we need to work on very much going forward. So this is basically an overview of how the committee structured the governance framework, and I'm going to talk through these as quickly as possible and go into a bit more detail about some and not others. I do want to say a bit about the ethical values and principles, and I really think this is important because the framework recognises that policies governing these types of technologies will likely vary from country to country. Nonetheless, the framework calls on all countries to incorporate these key values and principles into their chosen policies. It's not prescriptive of what the policies are or the tools countries need to use, but it is very firm on the need for these key values. And a lot of them you will recognise from the H3Africa Consortium and the work of the ethics working group. Looking at a lot of these of fairness, inclusiveness, social justice, but also really looking at some of these substantive and procedural, looking at the procedural ones, looking at the wider systemic issues about responsible stewardship of science. What kind of collaborations do you want to build? What kind of projects were really important? And I do think that these values and principles were a critical part of the committee's work. You will see they flow the whole way through the governance framework. They are embedded in all the case studies, in all the tools, but they are also through the recommendations. And I think they are an important step forward in the way that WHO is working in this space, and also in terms of how to translate ethical principles into policy and practice going forward. Again, I want to emphasise that at the heart of all of these are issues of equity that we are all grappling with at the moment. This is the special challenge. These are the areas I wanted to just point out that the committee looked at, but I will not go into this in great detail at this stage. I think this is a really important part of the framework, and this is really outlining the range of policy tools that might be used for governance, which include not only domestic regulation and judicial rulings, but also international instruments, institutional standards, funding conditions, professional guidelines, and patenting licensing provisions, among others. These are relevant for human genome editing. I think these will also be relative for genomics technologies moving forward. It really emphasises the need to develop these tools with input from a broad and inclusive range of interested parties. But the other thing I really want to point out is it highlights the need for not a narrow view of governance and oversight. It cannot just be the responsibility of one party. It must be about all the players in the system taking responsibility, whether you be the researcher, the research funder, whether you be the Ministry of Health. It's about different levels of responsibility and accountability. And it's also about collaboration, working together with all the different parts. And that is part of the ethos of responsible scientific stewardship. As I said, scenarios were a key part of the work of the committee. They felt it's really important to develop these to offer possible tools to a variety of scenarios in which human genome editing is offered in research and clinical settings, as examples of how the framework might be used in practice as policies are developed around the globe. And you'll see we developed 7 of these covering the following areas. And I've highlighted the one for sickle cell disease because I think this is live at the moment for the H3Africa Consortium and something that we could work on together in terms of implementation, issues of scalability and the types of governance, ethical oversight that will need to be put in place to make these clinical trials firstly go ahead and then the technologies available in an equitable and fair manner. So very keen to have further discussions with the consortium on that. I'm going to move now, as I said, it's a bit of a whistle stop. I'm going to move to the recommendations section. And I think when the committee was progressing, it decided very early on that a framework approach would be perfect for governance. But it was also alerted to real opportunities and challenges that went beyond just the development of the governance framework. And this is not just issues that they thought were important in terms of human genome editing, but more systemic issues, cultural issues, and issues that are really important for emerging technology more broadly. I think there's a really important point to make when developing oversight mechanisms, that there's becoming a blurring of technologies. You know, AI will be used with genomics. So it's really important when you make recommendations or you look at future governance mechanisms that you focus on things that are not just narrow in perspective, but can really take in that broad view. And this is really the ethos behind the recommendations. As you'll see, there are 9 recommendations areas, discrete areas. I'm not going to have the time to speak about all of them. I want to highlight a couple of them that I think are particularly pertinent to the H3Africa initiative going forward. I mean, I think they all are in one way or another. I just don't have the time to speak to them. So I'll pick up on a couple of them. So I'm going to skip the leadership one, but I think that it's important just to make the point that I think WHO does have a leadership role to play in embedding genomics in health benefits going forward. And the DG could do some of the same work that he's done in COVID for genomics going forward. So that was really the ethos behind the first recommendation. The second one I think is very fitting for the H3A collaboration or initiative, because it is about international collaboration for effective governance and oversight, and really does look at how we all work together and the different, what I'm going to say, mechanisms and approaches for regulation and up-regulating and down-regulating depending on the settings in which you work. So initiatives like H3A that have been at the forefront of setting oversight mechanisms when there may have been an absence are critical for us to learn from in terms of international and regional collaboration. So I think there's a lot H3Africa could take forward into effective governance and oversight going forward. As I mentioned earlier, one of the committee's early outputs was in terms of transparency and the importance of participants knowing what type of research was out there, was the establishment of the Human Genome Editing Registry. And I think for genomics technologies, there's some lessons to be learned from that, and we could build on that going forward. International research and medical travel, this came up a lot in different contexts. Often referred to now, I think, as this ethics dumping. And that is, this doesn't apply to all international research, but specifically where you're intentionally exporting research for one to another jurisdiction, which has lower regulatory standards in an attempt to evade stricter regulations. And the committee really wanted to focus on that and come up with positive recommendations for how we could look to deal with that going forward. And that is something that I think H3A in terms of ethics dumping and its collaborative model and the way that you've set up research collaborations is a good way that we could learn for emerging technologies going forward. The fifth area is what's broadly called illegal, unregistered, unethical, or unsafe research and other activities. And some committee members and some would refer to this as the whistleblowing. And this is really knowing areas of misconduct and seeing some of that research coming down the horizon, and how can we make mechanisms and put them in place to try prevent those. And this is something that I think does need a collaborative approach, but an entity like WHO at least could take a lead on starting it. But it'd be interesting to hear about H3 Africa's experience in this, or if it's had any experience, or what kind of mechanisms and models it's putting in place for this type of activity. Intellectual property, a huge topic, I think, a huge topic in COVID at the moment. And it came up in many of our discussions, and we had a specific webinar on this. And I think this will be a key one to build on the TRIPS agreements, the COVID discussions, and to work with WTO and WIPO going forward to look at this. But I think in terms of your work in genomic technologies, helping look at patent holders to encourage equitable access will be key to equity issues going forward. And then another key and this is the final one I really focus on, is the education, engagement and empowerment. And it's critical that we called it the 3 E's, and it really is important that you put the education at the beginning. And I know there's lots of valuable lessons from H3 Africa and the cartoons and the comics that you produced in terms of education. And I think there's a lot for us to do and build on what you've done in the African context for genomics. But this really does need to be something that's well coordinated, and we do need to start with the education, and not just of populations or the general population, but policymakers as well, and those in decision making, as to the value of these types of technologies, also to what are the potential benefits, but the potential harms. And I think that will lead to empowerment and better policies created, because they will be built in by knowledgeable people who have an interest in holding these discussions. So there are lots of great models out there, but I do think there needs to be a more coordinated approach to this. And then I'm getting very close to my time, so I'm going to come to the future look that I wanted to discuss. And this is really about WHO and genomics. And I'm pleased to say that when the science division was created a couple of years ago, not long after that, there was considered an opportunity to move the genomics portfolio. So it has been moved to the science division. And I think that's great, because it allows us an opportunity to really review it and refocus it going forward. Gives us an opportunity to expand our collaborating centre network, build collaborating centres in Africa, build on the great H3A bio network as well. And also, as I said, we can't look at things in isolation. So the Science Division has a real focus on emerging technology. So, for example, at the same time we released the human genome editing, we had a sister report coming out of the same department on AI and health. And that's really great that we can sort of bring all these pieces of work together. So I think that's an opportunity for WHO going forward. In April this year, the DG announced the establishment of the Science Council, to provide guidance on the science and research strategy of the organisation. And this will be a seminal council, very high profile, and it has interestingly chosen genomics as its first area of work. So again, I think this is a great opportunity. It's going to start with 3 collaborative or 3 workshops looking at the areas that I've listed on the slide. So looking at what are the current genomic technologies, or those coming down, and the potential applications, and where can they be applied, and what would be some of the challenges to applying them, some of the access issues, the training issues, the technical issues. And I'm sure these are all things that you are grappling with on the ground now. So there's a lot that we can learn from you. And then the third one is looking at the broader context of some of the ethical, legal, regulatory and related. So I'm very happy to give further information on these going forward. These will happen in the next coming months. And I think it's a great opportunity for WHO to collaborate with H3 Africa. And I'm going to conclude by saying I do think there's a real opportunity to engage H3A going forward. I just listed some key pieces of work, but I think there's a real opportunity for us to learn from H3 Africa, and that's what I really want to point out. And I remember many meetings that I attended, consortium meetings, and this phrase has always stuck with me, and I think this is key to the work that we've done to date. This go together, this collaboration, and I think it's key to our work going forward. So I want to thank you all for the opportunity, and I look forward to the Q&A session. Thank you.
Thank you, Catherine, for that fascinating talk and the great work that you're doing with the WHO. And it's nice to see you again. So there's one question from, we've got time for one quick question. There's one from your colleague, Audrey Duncanson, and she's saying, lovely to see you as well. You've described a major piece of work, very impressive. How has it been received globally, and has there been any shift in practice?
Thank you, Audrey, for the question. So it was launched at the end of June, and given COVID, really well received. I think a lot of people are starting now to think about these emerging technologies, especially the sickle cell community. So on the whole, very well received. We are currently doing the implementation strategy and plans now. We haven't, as it was only released then, really gotten into it, but I'm very happy and would love to engage, as I said throughout the presentation, with the H3A Consortium on some of the challenges that you are having with some of the trials and the studies you're doing and some of the things you foresee. So very happy to work with the community.
Great, thanks, Catherine. And I'm sure the H3Africa community will be really pleased to get involved. So in the interest of time, unfortunately, we have to move on now to our next speaker. So thanks very much again, Catherine. So Alex Shalik is our next speaker. He's the Pfizer Lobach Career Development Associate Professor at MIT. He's a core member of the Institute of Medical Engineering and Science. And he's also associated with many other institutes like the Broad Institute. And he's going to talk to us today about equity in the human cell atlas. Thanks, Alex.
Just let me know if I get close to time. I don't have a timer here. So thank you so much for the opportunity to present some of the work that's going on in the Human Cell Atlas, and particularly the work with respect to equity that I think will build on a number of the themes that just showed up in Catherine's talk. And I'm speaking on behalf of myself, Musa, and Partha, who are the co-leaders of the HCA Equity Working Group. And so just to sort of set the stage, I figured I'd tell you what the Human Cell Atlas is, and then I'd give you a sense for why we think it's important, and then talk to you about how we're hoping to engage, empower, and educate individuals to become participants, generators, and users of this data. So fundamentally, what the Human Cell Atlas is it's a desire to create a periodic table of our cells. I'm originally trained as a chemist and a physicist, so the idea of having a periodic table, a fundamental set of elements from which we can work, is incredibly attractive. The idea is that we'd like to have a comprehensive reference map of the types and properties of all of the cells in our body as a basis for understanding, diagnosing, monitoring, and treating health and disease. Now, why is the Human Cell Atlas important? Well, there are a number of different reasons that I could give you. I'll just throw a couple here, and then I'll dive into one in specific. You can imagine that understanding our cells, their properties, and their development could have untold potential in regenerative biology, in understanding how specific genetic variants might impact pharmacodynamics, in diagnostic applications, in thinking about drug efficacy and resistance or toxicities, in doing drug discovery, and critically, given the fact that I'm speaking to you from Boston as opposed to actually having the opportunity to speak in person, in disease mechanisms. And so just to give you a sense of this, I figured I'd dive into one example quickly to show you what the HCA is, what the data is, and how it's actually empowering sort of an understanding of disease biology. As I don't think I have to tell anybody, obviously, COVID-19 is a global pandemic. There have been over 200 million cases at this point, millions of deaths, and ongoing active searches, including here in Boston right now. Sort of early in the pandemic, it's very clear what some of the clinical properties of the disease were. In general, upper respiratory tract problems, including cough, as well as GI symptoms, and then some general symptoms that were hard to describe, like fever and malaise. Through some incredible work that happened across labs all around the world early in the pandemic, we got sort of the basic biology of the SARS-CoV-2 virus that causes COVID-19, where we found that it binds to this ACE2 receptor on the surface of cells, helped by accessory proteases, primarily TMPRSS2, infects cells, goes in and creates RNA, replicates, and then can create new virus. And so on one hand, we understood what the symptoms were at a broad level, and on the other hand, we sort of knew what was happening at a cellular level. But a big question sort of was happening at the beginning of the pandemic is really trying to understand what are the cells that are targeted by this virus and to what effect, and could we try and use that information as a way of developing better therapeutics and prophylactics. So this is a place where a lot of us thought that some of the data that we have been generating in the context of the Human Cell Atlas might be incredibly helpful. And that's because what the Human Cell Atlas is, at its foundation, is these large data sets of genomic information on individual cells, where we have expression, for example, of all 20,000 genes across many different cells, across many different tissues and species. And we thought that this might help us figure out what were the likely targets of the virus, and therefore begin to understand some of the biology that we were seeing, at a more systems level. So this on the left is a non-human primate. I'm showing you 12 different tissues. And on the right, I'm showing you its single cell representation. I want to actually dive into what this data is and how it's generated so that we're all on the same page. So basically what happens in some of the technologies that are foundational to the Human Cell Atlas is that, we take tissues, we cellularize them, so we break them down into their individual components. We then collect those cells using a number of different isolation techniques that allow us to isolate individual cells so that we can process their genetic material and do many of the things that you probably think about in bulk, but on a single cell level. So for example, RNA sequencing. So here, for example, is a device that my lab helped create that enables us to isolate individual cells, capture their RNA, and then process it moving forward. And so what we can do is we can isolate a number of cells, grab messenger RNA, just like you would in a standard RNA-seq experiment using oligo-DT primers, amplify, sequence, and then if we run this across, say, 100,000 cells and look at information on all 30,000 genes, we get this huge amount of data, going back to some of the points that Catherine brought up before about AI and analysis, from which we can begin to understand some of the underlying structures in that biology. And so here's just a video showing you some of the cool computational things that happen where we take in this large data set, filter for quality, identify genes that we care about, and then create plots where we try and bring cells close to one another that are similar in high dimensional gene expression space and push ones apart that are further apart. And I've colored this map by the tissue that I'm coming from, and you basically are watching this algorithm iterate and try and figure out who is like who, very much like a social network. So to give you a sense of what's actually behind this beautiful hairball, what this is each individual point, as I'm showing you on the right, is a single cell. And what that represents is this sort of vector of cell identity where we have information on the expression of all 30,000 genes within that cell. By looking at what genes are expressed, we can begin to identify the different populations in the target tissues in which they exist. So microglia, primarily in the brain, immune cells like eosinophils showing up in multiple different tissues, and then, for example, intestinal cells showing up primarily in the gut. What we can now do, if we want to begin to ask ourselves what SARS-CoV-2 might be targeting, is we can look for which cells have ACE2 and TMPRSS2, even without doing new experiments. So we can actually reuse some of this existing data to get insights into the current pandemic. And so in that example that I was just showing you right there, we can see the tissue that has the highest expression of ACE2 is the ileum. There's also high expression the jejunum and in the lung, in line with the fact that you see GI symptoms in lung infection. We can dive in a little bit more, and here I'm showing you some data that we helped generate with Al Leslie and Henry Cloverpris at RI in Durban, South Africa from human lung, where we've now gotten a little bit more granular, and I'm showing you sort of a similar plot with individual cell types being points colored by identity. We can ask ourselves, among these different cells that show up in the lung, which ones are our likely targets? And what we find is that actually ACE2 is expressed in a small number of cells, primarily ciliate cells and type 2 pneumocytes, and that the accessory protease TMPRSS2 is really in those cells as well. So to summarize a lot of work, what our data told us in going back and looking at some of this information is that in the nose, it was probably secretory goblet cells and ciliated epithelial cells. In the lung, type 2 pneumocytes and ciliated epithelial cells, and in the small intestine, absorptive enterocytes. The interesting thing from some of these initial looks at the data was that there's actually very few cells that are ACE2 positive and even fewer that are ACE2 TMPRSS2 positive, which didn't really line up with some of the symptomology of the infection. And so it was strange to see these needles in the haystack. And you might ask yourself, well, this is just using existing data. Can you use the same stuff to actually go in and move from a reference to figuring out what the actual biology of disease is? And the answer is yes. I won't dwell on this too much, but these technologies, because they work with incredibly low cellular input, can be used on just about anything. So here, for example, is a study that we recently published with some people from the University of Mississippi Medical Center and Boston Children's, where we took the same nasopharyngeal swabs that you maybe have experienced yourself, even though they're unpleasant, that are used to look for SARS-CoV-2. And rather than just using them to detect virus, we actually took some of the cells that slough off when they sort of brush around in your nose and went and did the same sort of sequencing on top of them. I don't have a lot of time to tell you about this. I'm happy to dive in questions or offline. Please feel free to contact me at any point. It's just shaylik@MIT. But we can sort of get an idea of what's happening in the epithelia, what are some of the underlying immune cells, and then share this data with the community. And there's lots of interesting biology here, some changes specifically in the epithelial compartment associated with COVID infection, and an ability also to identify which cells actively harbor virus and some of the factors that are associated with it. And interestingly, we found that actually, there's a change in innate interferon responses in epithelial cells that seems to be important for severe COVID. So I know that was fast, and what I started to show you is some of the data that's been generated using some of these technologies that let you look at messenger RNA or the transcriptome of individual cells. But in parallel, there have been sort of these incredible advances over the past 10 years, spurred by molecular biology advances and by advances in micro and nanotechnologies that now enable us to look at the properties of individual cells at unprecedented resolution, whether it's mRNA, chromatin accessibility, surface proteins, and the like. And really, this is what the Human Cell Atlas is trying to capitalize on. We're trying to take many different tissues, understand what are the fundamental cells that exist, what are their properties in terms of DNA, epigenome, RNA, to create these fundamental references, and then to use them as a basis for understanding, diagnosing, monitoring, and treating health and disease. So the HCA, has been building these atlases across tissues. I showed you some from a non-human primate and then a little bit from human lung, but there are many different atlases that are being actively generated across multiple different tissue compartments and stages of life. The HCA is a relatively nascent organization. Really the kickoff meeting was the Wellcome Trust in 2016 in October. And then there's been a whirlwind of activity, including computational jamborees, face-to-face meetings, focus meetings on very specific things like ethics or equity, meetings all around the world, for example, some of the more recent ones in Latin America, Asia. But obviously a little bit of this has been disrupted by the pandemic. I'll say that although it is young, there is a lot of committed funding to this, with a number of individuals supporting this work and its implementation. This really is a global effort. This is grassroots. There are many people around the world who are engaged. If you look online, it says that there's over 2,000 members, but there are many more than that that aren't signed up. Within Africa alone, there's 12 countries, 23 institutes, and 25 members engaged, and that's great, but it really is not where we'd like it to be. We'd really like to have many more people involved, many more people creating data, many more people actually defining the direction in which the HCA is going. Since, as I said, this really is grassroots. I think that what's key and what I wanted to emphasize here today is that the HCA is something that has always been anchored on egalitarian and comprehensive principles. We've always wanted to be computationally inclusive, even from the get-go, considering gender, age, ethnicity, tradition, culture, disease, really create a cellular atlas that's representative of humanity so that sort of this fundamental understanding of health and disease could be applied to the problems that face all of humanity as opposed to a specific sector. Organizationally, this is grassroots. We'd love to span countries, levels of education, get involved in training of researchers. And really, for this, we've been spending a lot of time having conversations with people trying to identify barriers to success so that we can get around them. And, we've wanted to touch all sectors of society. I liked when Catherine talked about this. We feel the need to educate and be educated by everybody that would be involved. That's essential to the generation of this, not just scientists, but, public, people involved in government and administration. Here I'm talking to you about equity. And as I said, this is something that we talk about even from the initial meeting at the Wellcome, where Mr. Parth and I worked there. We had an initial strategy meeting back in June of 2019, which feels like a lifetime ago before the pandemic. We really tried to shape and articulate what our goals were and develop an initial action plan. And then, later in 2019, we had a meeting on the shoulders of the Grand Challenges meeting in Addis Ababa, where we more formally recommended compositional, organizational, and educational goals for the HCA. I think that really, it's important to start by saying, what do we mean by equity and how do we define it? And for us, what equity really means in the HCA is inclusive representation, participation, and benefit from data. And so when we talk about representation, we really mean that we want the data that we're generating to represent, a diversity of sexes, ages, ethnicities, geographies, and socioeconomic statuses. When we talk about participation, we mean that we want to foster capability and leadership in low and middle income countries. We want everybody to be involved in this. We don't want this to be something that is creating, a greater gap in sort of genomic technologies and in information. And we want to make sure that we're getting equal benefit from the data. I think that in a lot of places, there's a tendency to make data, and then there's a question of, well, can everybody take advantage of it? And really what we want to do is we want to make sure that we can overcome existing barriers and learn from others so that we can avoid past mistakes. We don't know all of them, but we'd love to have conversations so that we can make sure that, we do the best by everybody who's engaging this as we possibly can. So I think that, those are some 3 basic principles, but as we think more holistically about this, key things that we think about are empowerment, shared sovereignty, open participation, engagement, shared opportunity, transparency, fair funding. And I'm happy to dive into any of these, but I think core to what we've realized is sort of, similar to the 3's that came up before, is that really what we need to focus on is empowerment, education. I call it training and outreach. And so, before the pandemic, we really tried to focus on this. And, there were a number of different things that have already happened within the HCA. I told you about the initial strategy meeting in London, where people from all around the world came together to start to talk about the HCA. There have been a number of roadshows that have focused on going and talking about the HCA, introducing technologies, starting to talk about computational methods, building local leadership. And I show you, for example, some of the work that we did down in Sao Paulo, Brazil, as well as some snapshots from the initial meeting in Addis. We have some success stories, even though we're young, but we have a long way to go. We have a diverse working group, and we're working to build networks around the world. We've spent a lot of time building capacity and creating leadership and partnerships within Latin America. And now have multiple different countries and investigators involved, and sort of centers that are driving a lot of the science of local relevance, particularly in Brazil, and helped to raise some local funding. And we've done a lot of early outreach in other countries, engaging in the MENA region, in India, and in parts of Asia as well. One of the big things that we've been doing in this, our roadshows, has primarily been in person. We've done a couple remotely as of late. We saw that going to Brazil and engaging with scientists there, and learning about local priorities and helping them figure out how we could work with them to solve the things that they wanted to solve instead of the things that we thought were important, was incredibly powerful. And so we've had plans to do roadshows in Vietnam, in India, and tentatively in Ghana. We were very advanced actually and ready to go to Vietnam right when COVID hit, and so that's been delayed. But we're looking to schedule these. We've been talking about doing, as I said, a West Africa HCA initiative in Ghana. We'd love to do many different engagement opportunities and get researchers involved all around Africa. And we're just beginning some planning activities in India and Bangladesh. We've been doing, I'm about to be done. We're working on a ton of virtual trainings and resources, and would love to talk to you about that. And we've shared some of the lessons that we have in this commentary that we wrote up. I just say, we're thinking about how to work with partners to ensure sampling. We're working with the, to deal with complexities, and we'd love to hear what the problems are so we can help solve them. And we're working to, across sectors to try and get buy-in where necessary. I'll say that, we're doing workshops. We're trying to do travel grants to enable people to go back and forth, and trying to create seed grants so that people can work together. And then finally, I'd say, really success to us is getting a number of people involved from all around the world as data generators and users. And we're really trying to track and report that so that we can figure out whether or not we are becoming more equitable. There are lots of ways you can help, but I'll just say, please contact me. I'd love to chat with you. And thank you for your time and partnership, and sorry for running a couple minutes over.
Thanks, Alex, for those interesting insights into the Human Cell Atlas, and also the amazing work that you're doing in trying to get other people involved in the equity side of things. So we have time for, we've kind of run out of time, but I'm going to give time for one quick question. So there's one from Ashley Bemiku. She's saying, interesting work, Alex. How does the system accommodate the dynamic nature of cells as they encounter different stimuli, like pathogens or environmental changes?
So it's a fantastic question. I talked about one technology, which is single cell RNA sequencing, which is sort of this technology where you lyse cells, so you freeze them at a particular point. So in many places, we do time series so that we can study sort of the dynamics of that interaction. But there are other technologies being developed that enable you to do dynamic tracking, match with endpoints, or some more complicated pieces there. But for this, think of it as it's a snapshot. One of the interesting things about the snapshots is that not everything is synchronized. So you can actually infer dynamics through these pseudo-temporal methods, where you can infer time even from one look, just because not everybody is at exactly the same time point. But there's more work to be done on sort of dynamic live tracking. And I'd be happy to chat about some of the technologies that are coming online and actually how they're being used to study some of the things you're asking about, specifically environmental perturbations and pathogens.
Thank you very much, and thanks again for your talk. So Deo, please keep an eye on the Q&A, because if people want to post additional questions, then you can also go there and answer the questions there. And please do get in touch with Alex if you want to hear more about it. And for the audience, please do remember to post your questions on that live Q&A. So thanks again to both our invited speakers. And now we're moving on to report backs from some of our projects. So the first will be from Kofi Ani, who's part of the H3 Africa project, who the PI is Professor Solomon Afuriakwa, and he's going to be talking about clinical phenotypes and ethical considerations for return of results. Thanks, Kofi.
Thanks, Nikki, and thanks to the organizers and my group for Okay, so I'm presenting on behalf of the H3 Africa Network. And my presentation will cover two main areas. I'll be looking at clinical phenotypes from the point of view of the laboratory core, which is one component of the H3 Africa project, and also Project 3, which is a cardiovascular project. Under the ethical considerations, this comes under the community engagement core that I lead. So just as a quick background, in terms of H3 Africa, is that severe hemolytic anemia is a hallmark of sickle cell disease. That is something that goes on all the time. Although from the patient's point of view, they tend to focus on things like pain and other symptoms such as fatigue, but which have the underlying cause being this hemolysis. So also toxic molecules from hemolysis acting as danger-associated molecular pattern damp molecules cause tissue injury and ultimately end-stage organ damage. And that is what we are interested in studying. Nonetheless, natural defenses against hemolysis and organ damage are poorly understood. So in terms of the objectives, we have three main projects. Project 1 is basically a GWAS to identify genetic markers of 5 hemolysis cytoprotective proteins that neutralize these damp molecules and cause acute organ damage. These 5 cytoproteins are haptoglobin, hemopexin, alpha-1 microglobulin, heme oxygen 1, and ferritin. Project 2 shifted from a human project to an ex in vivo, and it's now looking at how we can determine the influence of hemoglobin variants on the sexual life cycle of Plasmodium falciparum, which is a malaria parasite, mosquito parasite, in terms of asexual as well as sexual reproduction. Project 3 is to determine whether the cytoprotective proteins are associated with specific echocardiovascular phenotypes. Our study sites are 6, 2 in Ghana, 3 in Nigeria, and 1 in Tanzania. So just as a update from the lab core, the lab core actually has a biorepository in Accra. This is not the H3 Africa biorepository. This is the initial storage facility where all the samples from the 6 sites are taken for preparation before they will be ultimately moved to the H3Africa biorepository, which is based in Abuja. Now, looking at the samples, so far we have 5,741, 2,320 adults, 3,221 children. And as you can see, the majority of children are from one site, which is the Kumasi site in Ghana. And also the majority of adults are from the Accra site in Ghana. And in terms of in terms of adults, we don't have any sort of adults at the Kumasi site. So that is the only site that hasn't got any adult participants or patients. Now, the initial work that the lab core has been doing includes hemoglobin electrophoresis. And this is basically to confirm the diagnosis of the participants, because obviously in terms of the studies, we are looking for people with sickle cell disease. And interestingly, from that initial work, approximately 3% of the participants do not actually have sickle cell disease. I mean, some of them actually have traits, either AS or AC traits. Some of them also have CC disease, which is basically not sickle cell disease. But the interesting thing is that 33 participants, that is 0.6%, actually have hemoglobin AA, which is basically that they actually just normal, they don't have a trait or they don't have sickle cell disease. So this was quite interesting and has to be obviously fed back to the participants, because what we have is that the participants are actually recruited from sickle cell clinics. So if some people have been misdiagnosed as to not having sickle cell disease, what exactly are they doing in a sickle cell clinic? And that is quite important from an ethical point of view. So going on straight to project 3, which is, as I said, an echocardiovascular project. This project has a target of 2,000 participants, 1,000 to be recruited from Kano, and then another 1,000 to be recruited from the Accra site. And unfortunately, because of COVID, there's been interruptions, and the Accra site is now sort of about to start recruitment. That was supposed to be after the Kano site, but the Kano site was delayed, some equipment issues, and as well as COVID. So at the moment, 822 participants have had their echocardiogram, which relates roughly to 31% males and then 69% females, which is a 1:2 sort of ratio. And again, the mean age is about 23 years, standard deviation 5.8, and going from a range of 18 years to about 55. The mean systolic blood pressure 116 with a range of 65 to 181, and the mean diastolic blood pressure 71 or 72 thereabouts, ranging from 40 to 126, and a heart rate of about 86, ranging from about 56 to 135. Another important indicator of, let's say, heart problems or cardiovascular dysfunction is the TR jet velocity, which is basically measures kind of a flow. And anybody who has a TR jet velocity of about 3 tends to have, actually 2.5 tends to have cardiovascular dysfunction, and above that, 3 quite severe. So the majority of the participants, about 70%, appear to be okay, and then remaining about 30% had an indication of some problems. And then going to the left ventricular systolic and diastolic functions again, the majority, 95% again, indicated greater than sort of equal or greater than 55%. Might show some problems with the ejection fraction, and the majority, 97% actually seem to be okay. Same with the diastolic function, which is again, those showing some sort of normality are the highest in proportion, almost 66%, but in impaired sort of relaxation and also restrictive filling is also quite high from that point of view. And again, also looking at another function, which is the E function, again, over 15% would be kind of problematic. And you have, 15% did not indicate any problems, and over 15% was, which indicates some sort of restriction in terms of function, is about 18%. So just to summarize quickly, about 19% of the participants have abnormal left ventricular geometry, for which 84% of whom also have an index of dilatation and remodeling. 3% have left ventricular systolic dysfunction, which is not a huge number, but important. And then 18% also indicated possible systolic dysfunction. And only 2 patients had an EE greater than 15, which is, again, a very small number. In terms of the TRV, 44% had, a TRV velocity, but 83% out of that indicated that they were mild, 60% moderate, and 26% had a TRV velocity over 2.5. So basically, when you're doing the echo, some patients would have a TRV velocity. Some patients wouldn't have any indication of that on the echo. But if they do, then anything greater than 2.5, as I said earlier, might indicate some problems. And in this case, 26% had a velocity over 2.5%, for which only 4.2% had greater than 3, which is that indication of suspected dysfunction. And then only 3 patients also had some problems with the right ventricular systolic function and RV fraction area change of less than 35%. So that was quite small in terms of the numbers and indications. So moving on then to the ethical considerations in terms of what the framework that we used community engagement initially to work through some of these ethical issues. We focused on 3 main areas. That is autonomy in terms of research, decision-making, and participation, specifically in sickle cell Africa. We looked at best interest in terms of what participants might want versus what we, let's say, researchers might want from participants from a genomics point of view, and trying to address that. And then also importantly, from the point of view of let's say, healthcare services initially not being available at the right levels from the point of view of the patients or the participants, but also what duty of care we have to be able to offer improved or adequate sickle cell services by engaging also with health decision makers or policy makers who are representatives of the government. And we did publish our initial findings in the focus groups earlier on. But what I wanted to talk about a little bit today is also the participant survey that was the next step. So this was after the participants were actually recruited into the Sickle Gen Africa studies. The community engagement was initial. That was before patient recruitment took place so that we could address some of those issues. But this was afterwards. And the focus was trying to look a bit more about research, participation, decision-making. That is in terms of how participants or patients would make decisions about research, whether in terms of autonomy or in terms of their best interest, and trying to find ways in which we could use that as a means of understanding, sort of non-participation better, but also trying to address any ethical issues that might be relevant. And then the second area that is also very important and topical, especially within our discussions in H3 Africa, is to do with the return of individual results, as well as incidental findings that might have some kind of impact or health impact from the point of view of the participants when there's actually no treatment available at the time. So looking at some of the initial analysis, the results, we had 624 participants. Our target was 600 with approximately 100 participants from each site, which we're able to achieve, but with the Lagos site having quite a number that is much higher than the target, which was good. And again, in terms of the male-female ratio, it looks like, again, it's about 1/3 male and 2/3 female from the participants of that survey. And looking at the research decision-making issue, we try to find out how participants would make decisions, especially in terms of their health generally around sickle cell disease, and also in terms of research participation when it comes to sickle cell research. And by looking at whether they do consult others or they take the decisions by themselves. And as you can see, the majority would consult others. And these others would include people like their spouses, community leaders, religious leaders especially, and also other significant people that they would consider, which may include other family members. And interestingly, from the point of view of whether they would be interested or they would have a preference in terms of the return of individual results relating to possible risk factors associated with organ damage from let's say the GWAS studies, for example, and incidental findings that may also appear from the genomics that we might be able to report back. They seem to be overwhelmingly in the majority, 98%, with only 1% saying no to both and 1% saying that they were undecided as well, which is quite significant. Yeah. In summary, the Sickle Gen Africa has the largest cohort of sickle cell disease patients in the world. We have a target of 7,000. We are close to that. The majority of participants are in favor of return of individual results and incidental findings, regardless of treatment availability. Nonetheless, they did indicate that patient support is required for feedback, which includes genetic counseling and some psychosocial support if they are to receive bad news. And just to acknowledge PI, Solomon Oferiakwa, multiple PIs, Gordon, Awandari, Julie, Makani. Also, all the study participants so far have been very, very good and great. And this is a whole bunch of people who are part of the network. Thank you.
Thank you, Kofi. Again, fascinating talk. So we've only got time for one quick question. So we have one from Kutala, who's asked, may I please ask about the participants that were misdiagnosed by the SCD clinic? Were they showing any clinical phenotype of SCD, or they went for diagnosis just to know the SCD status?
So they are probably more likely to have been diagnosed outside the clinic, because they are, like in Africa and a lot of these countries where we don't have very good laboratory services, people just go to, places where they can have their sickle cell status tested, which is not necessarily electrophoresis. But the key point is that people tend to present in the clinics with symptoms. So if you don't actually have a confirmatory diagnosis, you would just assume that they have sickle cell, and then you sort of enroll them in the clinic, and that's what tends to happen.
Thank you. And there's another question from Jennifer Troyer, but perhaps you can answer it in the Q&A as we run out of time. So thanks again for your talk. So now we'll move on to the next set of presentations, and these are about the first set of presentations about the bioinformatics training grants. So the PI is Dan Masiga, and our first speaker is Suhaila Hashim, who's going to be talking about EANBIT from the Pwani University perspective, and then we'll go straight on to the fellows presentation from Shahib Kuyaga on genetic diversity, distribution, and characterization of clinical Pseudomonas aeruginosa strains from Kenya. So, and then we'll have a live Q&A, a joint Q&A after that. So thanks, Suhaila.
Good afternoon, everyone. Thank you, Nikki, for the invitation. I'm Suhaila, and I'll be talking about EANBIT and how it has impacted Pwani University thus far. So the objective of EANBIT has been to provide a curriculum, an MSc Bioinformatics curriculum that is harmonized and that can be adopted across the East African countries, the universities in this region. And the Pwani University is one of the partners. So a little bit about Pwani University. I've not been to Pwani. We are located along the Kenyan coast in Kilifi County, and we're right at the Indian Ocean. It was formerly an Institute of Agriculture established in 1984 after Regulatory University College in 2007, and finally got our charter and became a fully fledged university in 2013. So here are some images to show what it looks like. We have the main gate. So for those of you who are coming to visit, hopefully, we are right. So it's not easy to miss out the main gate. We have the administration block, which is equivalent to the White House, you could say. The university library, the botanical garden, and a huge farm. We sit on 600 acres of land. So we are a very young university, but we are very big in terms of size. So the academic division is organized into 8 schools: School of Agricultural Science, Education, Business and Economics, Health and Humanities and Social Sciences, and the pure. These 7 schools offer programs all the way from to diploma and PhD level, while the School of Graduate Studies normally coordinates post programs at Pwani University. So the MSc Bioinformatics program, which is funded by EANBIT, supported by it, is hosted at the Department of Biochemistry and Biotechnology, where I have been the former chair of the department. until February this year. The program is also supported with 2 other departments, biological sciences and mathematics and computer science, within the sciences. Also anchored at PUBRIC, which is the Pan University Biosciences Research Center, where we have infrastructure in terms of the lab, HPC server, and currently the manager of this research center. So ENBIT has played a very pivotal role in ensuring that Pan University and kick off the MSc Bioinformatics course, right from the curriculum development to inspection and accreditation by the Commission for University Education. And within the ENBIT network, Pan University was actually the first university to be given the green light to that program. So we were quite happy with that. In addition to infrastructure, you can see the bioinformatics lab on my left and the HPC server and security systems which were put in place thanks to ENBIT as well. In addition to supporting fellowships every year for 3 years, the last 3 in terms of tuition and stipends. This might sound really on the ground, it's very difficult for our students to be full-time students if they're self-sponsored. Really help to ensure that we get students for this program. And also facilitation for visiting lecturers. So we started out in 2018 with our cohort, and all of them completed by July 2021 and graduated, COVID notwithstanding. The second cohort, they are now currently doing their thesis of submission for examination. Then the third cohort, they've completed their coursework and they're now currently finishing their residential training, and we are just finalizing for the fourth cohort. So what does this all mean for Pan University? In Pan University, our vision is to be a world-class university in social, economic advancement. So the products, the graduates that we're producing from this fine course, informatics, that is funded by ENBIT, has resulted in producing graduates who have been absorbed internationally. You can see from John Okech, of his PhD at Leicester University, Michael Landy, who has also commenced his PhD, Maggie and Joseph, who are about to start their PhDs in Italy and Germany, and Martha, Luca, and Joy, who are already employed as data informaticians in their respective institutions. It's also not too far behind. We can start with Ruth Nanjala. She, through presenting in international and local conferences, they get to win awards and travel grants. I think Ruth last year she won one of the categories Africa Consortium meeting, the last one. Ivan Serwa extremely well. We're really proud of this cohort, and already he has 4 publications in journals from his master's work, and he has won a travel award to attend an advanced course in antimicrobial resistance at the Sanger Institute. And the cohort 2 are already also getting employment and PhD opportunities. For example, we have Winfred Katua, who's already secured a PhD scholarship with Wellcome Trust at the University of Bristol, and she's about to just submit her thesis and then already proceed for her PhD. So this is just a sample. By all means, the others also well. We are really, really proud of our students. And it is thanks to this network, the ENBIT, all our partners, starting from ECPE, where our PA is hosted, and the program coordinator, other collaborators, Camry Wellcome Trust, the IDEAL program, who also helps the bioinformatics course, the Becker ILRI Hub. And we have Uganda Virus Research Institute and Makerere University, and the Muhimbili University in Tanzania. And the network has expanded greatly, and we have University of Cape Town, Georgia, Tennessee, Nairobi University, and JKUAT. They help with research projects, supervision, thesis examination, and even adjunct faculty. We have a adjunct faculty all the way in Finland at Orion Pharmaceuticals. So with that, we really want to appreciate ENBIT, because it is through ENBIT that we are able to acquire this worldwide recognition and visibility at Pwani University, being a very young institute. I'd like to thank sincerely Dr. Dan Masiga, our PI of EN, attending this invitation to present today, and all our and collaborators, Pwani University Management, the CUE, our founders, NIH Bogati, all of whom have helped to make this MSc Bioinformatics course at Pwani University a huge success. And finally, thank you very much, H3Africa, for your support today. Thank you.
I'm Shahid Chiaga, a graduate fellow of the NBIT project, presenting genetic diversity distribution and characterization of clinical Pseudomonas aeruginosa strains from different geographical locations in Kenya. So Pseudomonas aeruginosa infections are usually multi-drug resistant infection, which is a challenge globally. And about 90% of the multi-drug resistant Pseudomonas aeruginosa globally belong to three major high-risk clones like ST111, as it's shown in world distribution and European distribution on the right. In Kenya specifically, a study at Aga Khan University Hospital in 2008 reported about 13.7% prevalence of multi-drug resistant strains of Pseudomonas aeruginosa. And these strains are usually associated with hospital-acquired infections. For example, a study in Kenyatta National Hospital shown about 54.8% of hospital-acquired infections associated with Pseudomonas aeruginosa, and 23.4% of hospital-acquired infections in Moi Referral Hospital also associated with Pseudomonas aeruginosa. These Pseudomonas aeruginosa strains contain extended spectrum beta-lactamases and carbapenemase enzymes, which usually confer resistance to penicillins, cephalosporins, and the carbapenem class antibiotic classes, which are commonly used in Kenya. So this antimicrobial resistance challenge globally and specifically in Kenya is a present threat. However, Pseudomonas aeruginosa strains from Kenya have remained scarcely explored through whole genome sequencing and also not identified by multi-local sequence typing. This information adds to genetic and epidemiological mapping to the global and local pictures of the distribution of this pathogen to combat antimicrobial resistance. As such, this study is to characterize Pseudomonas aeruginosa isolates from hospitals across Kenya and address the essential World Health Organization reporting measures of antimicrobial resistance. This is a cross-sectional study nested from one ongoing study, which is antimicrobial resistance surveillance in military and civilian populations in Kenya. The samples in this study were collected between 2015 and 2020 from patients in 9 hospitals in 5 counties across Kenya. That is Kilifi, Kisumu, Kisii, Kericho, and Nairobi. And the techniques being applied here is whole genome sequencing based on Illumina sequencing technology and Oxford Nanopower sequencing technology. So from our objective 1, which is identifying the diversity and distribution of Pseudomonas aeruginosa strains from the different hospitals across Kenya, we did a phylogenetic analysis and multi-local sequence type on our isolates. And the maximum likelihood phylogenetic analysis tree was built based on core genome SNPs in all isolates from different sources across Kenya. Also, we ran multi-local sequence typing, which is shown on the outer layer of the graph you are seeing, of the graphic you are seeing on the left. So in the graphic there, we identified about 48% of the isolates to be from Nairobi and 26% to be from Kisumu, among others. And out of all these isolates, 41 distinct sequence types were identified through multi-local sequence typing out of the 56 isolates. Out of these 41 sequence types, 9 isolates out of 56 actually had a sequence type 357, as you can see here. And also, one isolate from Kilifi also had the same sequence type. This actually shows that there was a near to an outbreak of multi-drug resistant Pseudomonas aeruginosa, as sequence type 357 is a high-risk multi-drug resistance clone. We also identified novel sequence types, which are shown by sequence types here with the stars on this picture. And these sequence types were sent to a Pabm LST, excuse me, they were sent to Pabm LST for curation and assignment, and they were assigned the new sequence types as are shown in this table below and this table. These sequence types were based on allelic profiles of housekeeping genes, which are usually 7 housekeeping genes that are based on to show diversity in Pseudomonas aeruginosa. Also, we had an objective where we looked at genetic relatedness of the Kenyan Pseudomonas aeruginosa isolates to the global isolates. Here, we did a phylogenetic analysis, and we created a maximum likelihood phylogenetic tree, which included reference genomes, global reference genomes like PA01, PA7, DK2, PA14, among others. So from what we can see here, the core genome phylogeny confirmed that our strains were across, were distinct strains across and spread across the 2 main groups, which are commonly known in Pseudomonas aeruginosa. Group 1, which is usually, which I marked as green, clustered as green, is a common phylogenetic cluster in Pseudomonas aeruginosa, most especially among clinical isolates, and it mainly contains reference genome PA01 and reference genome DK2. Group 2, which is usually the smaller clustering, which is above here, highlighted in pink, usually includes well-known virulence factors, virulence strains like PA14, which is around here, and then VRFTS04, which are commonly known to be multi-drug resistant. So we also had objective, which is determining the genomic characteristics, and we focused on virulence genes and microbial resistance genes. So for virulence genes, we had a maximum likelihood phylogenetic tree built on core genome SNPs, and also the presence and absence of virulence genes as detected from virulence factor database. And out of 156 genes identified, variation was found in virulence genes of about 34 virulence genes. This was also more evident for a set of effector virulence genes, which belong to type 2 secretion system, that is XOS and XOU, as you can see here. Most of the samples, they did not have XOU, but had XOS. And also that included most of our novel sequence types, actually all our novel sequence types. However, down here we can see that the sequence type 357, which is quite multi-drug resistant clone, it's also showing that here it does not have XOS virulence genes. However, it had specifically XOU virulence genes. And this sequence type also showed very minimal virulence genes across the virulence genes we got in variation. So this shows that actually most of this sequence type has minimal virulence. However, we shall see that it has a very high antimicrobial genes, high number of antimicrobial genes, as we can see here on this antimicrobial antibiotic resistance genes heat map. So here it shows that actually this sequence type of 357 has a high number, and they are the same resistance genes variation across. Also from unsusceptibility testing profile, we realized that actually we found it a multi-drug resistant sequence type or clone, because it was resistant to all the antibiotics that we tested on, as you can show on this, heat map here. We also realized that our novel sequence types actually had very few resistance genes, and all of them were non-multi-drug resistant, as you can see here. These resistance genes are basically associated on beta-lactamases, tetracyclines, sulfonamides, phenycols, fluoroquinolones, and macrolides, among other antibiotics drugs. These were isolated from CAD database. Thank you so much. I would like to acknowledge my supervisors, the AMR team I have worked with, and also ENBIT project, Makerere University, African Center of Excellence, Water Read Project in Kemri, Isipe, and Kemri as an institution. Thank you so much, and your questions are welcome.
Thank you very much to both the speakers. So I haven't seen any questions in the chat, but I have a question for Shahid. So you looked at antimicrobial resistance, but when you were looking at those virulence genes, presumably that's virulence generally, not just about antimicrobial resistance. For those ones where you found SNPs, was there any correlation perhaps with any clinical outcome from the patients from which those isolates were retrieved?
Thank you so much, Nikki. I hope you can hear me.
Yeah.
So what happened is that this study is actually a nested study where the mother study started over in 2015. So some of the information which is like clinical was part of totally different studies. So we didn't want to associate it much because of the ethical processes. So by the time we got to get the ethical approval, we tried to obscure the patient information to associate it so much. However, what we did is that we associated the virulence genes and the SNPs according to the regions where the isolates were associated, because we are looking at this in terms of Kenya as a country and the different regions in Kenya. And we realized that the virulence genes, for example, from the SNPs in the virulence genes from sequence type 357, which is quite multi-drug resistant, were actually very minimal, and yet they were almost all of them from Nairobi as a region, which is the capital of Kenya. However, when you associate that to antibiotic resistance genes, we actually found that any sequence type which had very minimal virulence genes, SNPs in virulence genes, had a very high number of SNPs in resistance genes, which brings us to a question of where a pathogen compensates resistance for virulence or virulence to resistance. Now, those are some of the questions we are trying to answer, because every time the virulence SNPs were low, the resistance SNPs were high, and vice versa. So that's the question we are trying to answer. Thank you.
Great, thank you. That's very interesting. So there are no additional questions on the chat, and we are running out of time. So please do look for questions on the discussion forum and the live chat. Please post questions to them from the live audience. So thank you very much again. And we're going to move on now to the next set of talks, which is related to the RHD-Gen project. So the first speaker will be Mark Engel, who's the PI of the project. And then there'll be a fellows talk from Tafadis Siwe Machapisa. And so we'll have those two talks, and then we'll have a joint Q&A session. Thank you.
Good afternoon, everybody. My name is Mark Engel, and we are really thankful to have this opportunity to be sharing with you the work of the RHD-Gen network. I will start the presentation, followed a little bit later by Tafadzwa, who is our RHD-Gen PhD fellow in the consortium. Now, rheumatic heart disease is the most common acquired heart disease in young people under 28 years of age. And it all starts with a strep throat infection. And there is debate as to whether it could also be a strep skin infection. But largely the understanding is that following a strep throat, after about a month or so, the body gives rise to these antibodies, which in a percentage of people erroneously recognize body structures as being foreign, and then, of course, attacks these structures. At this stage, the disease is called acute rheumatic fever. It's reversible and can involve the joints, it can involve the brain and so on, but most importantly, it involves the valves of the heart. If the infection continues, it could lead to permanent valve damage, which is irreversible, and it's at this stage that the disease is then called rheumatic heart disease. Now, around the world, we estimate that there are about 40 million individuals living with rheumatic heart disease, and globally we see about 300,000 deaths each year. The risk is higher in females, and we know that a lot of the females are picked up during pregnancy when echo scans are done of the heart. Rheumatic heart disease still needs a lot to be discovered in terms of the biology of the disease. Now, when one looks along the spectrum of the development of rheumatic heart disease, it does represent quite a few opportunities for intervention. And we speak about primordial prevention, where we look at issues relating to socioeconomic status. Primary prevention deals with making sure we treat the strep throat infection or the skin infection. And secondary prevention then refers to putting the patient on penicillin prophylaxis for 10 years, and hopefully that will stem the tide before the patient needs surgery, or worse still, succumbs to one of the effects shown there. Now, yes, we do understand this aspect of the work. However, there are even problems with addressing the disease at these intervention opportunities. Lacking, healthcare workers don't identify the strep throat properly, the patients are not compliant because the injections are painful, and of course surgery is very expensive and non-existent in many places. And this really then gives us the motivation for our study. When we look at the small studies that have been done, we see that there's a lack of robust genetic associations in these small studies. We know from a systematic review that we did not too long ago that there definitely is a heritability component. And in our case, we see that in Africa, we still have amongst the highest burden of rheumatic heart disease in certain populations. And there have been studies done elsewhere, but no studies yet in Africa. And so this really set a perfect opportunity for us then to tackle rheumatic heart disease from a genetic perspective, and thus set up the RH3Gen network. Many of you will remember Professor Bongani Mayosi, who really had the vision for this network. And we really wanted to do four things. We wanted to build a network for the phenotyping of rheumatic heart disease, to have an idea where the cases exist, to set up these connections with people working in the field. And from these collaborations then, we would then go on to identify the genetic variants affecting susceptibility and resistance. But in the process, we also wanted to make sure that we leave each of those collaborating centers in a better place, where we train local scientists, doctors, and set up research protocols and infrastructure so that indeed many will go on to investigate further diseases in the future. And then we also were very concerned to address the LC that was relevant to Africa, and many of you know Jantina de Vries, who did a lot of work in this area. So narrowing it down a little bit, specifically our aims and objectives were to identify the genetic variants for RHD susceptibility. We also wanted to identify genes and biological pathways involved, to determine the SNP chip-based heritability in Africans, and then to validate the GWAS findings using TDT testing. So how did we do this? We chose 8 sites from Africa. Many of these folks had been involved in a previous study, the Remedy study, where it was an observational study documenting the prevalence of rheumatic heart disease in Africa. And from these sites that were chosen, we then sought to recruit 3,000 adult cases and 3,000 ethnically matched controls. Each patient, each participant was characterized in terms of a number of parameters related to the heart. Obviously, in the controls, we wanted to make sure that they were free of a rheumatic heart disease. Echo was applied to all of these patients, and so we have all those data. Blood and DNA were collected, brought down to Cape Town, and then the genotyping was done in collaboration with our colleagues and friends at the PHRI in Canada. Okay, at this point, I'm going to hand over to Tafadzwa, who was based in Canada and was instrumental in the analysis and the work of the GWAS. Thank you. Over to you, Tafadzwa.
So basically, our GWAS results indicated that we had a chromosome 11 region that was genome-wide significant in Black Africans. You find that the minor allele frequency was common, and you find our odds ratio shows that this region increases the risk of RHD. Next slide. So basically, you'll see that this table is a summary of all of our top candidate SNPs, and you'll find that the chromosome 11 region in the Black Africans was the top SNP, and you also find that we also had other top SNPs in our admixed African individuals, which are what is commonly known as colored South Africans. And we also meta-analyzed these data sets, but we found no genome-wide significant regions in this area. Next. Replication of previous genetic associations were tested amongst 2 major data sets, the Oceanian data set, and we also tested the IGH locus that was previously found by Tom Parks. We replicated the previous genetic association from the IGH locus, and unfortunately, the other locus was not replicated. However, it was in the same direction of effect. But this still proved that our finding was true because it's been found in more than one population. Next. We also did some secondary analyses. Unfortunately, as is commonly known in Black African datasets, GTEx has mostly European samples. So when we looked for expression profiles in GTEx and in the GWAS catalog, we couldn't find our chromosome 11 lead region. However, we moved on to do pathway analyses, and we took a tool that is in silico called DEPICT. I've previously presented these results for DEPICT, and you'll find that cardiac valve tissue was the top region of expression, and this is consistent with biological data for RHD pathogenesis. Next slide. Our pathway analysis was also conducted with another tool called MAGMA. We were looking at which would be good candidate gene sets. You'll find that we had regions in chromosome 14, as well as other regions that have been seen in some previous proteomic studies. And you'll find that we had popular pathways, which include ECM disassembly and cardioblast differentiation. Next slide. We also did our heritability analysis to see if we could see around the same kind of genetic heritability we had seen from the twin studies. So in the twin studies from Engo et al. In 2013, he found 60% heritability. And from our study, from just our chip-based heritability, we found in Black Africans close to 50% heritability. In admixed Africans, we found 35% heritability, which shows that this is quite a highly heritable trait. Next slide. We also wanted to validate our findings because we had an independent trio data set that was collected from these same. This trio data set would include a parent and two parents and one child. And we did what is called a polygenic TDT. So this uses PRS and it leverages the transmission disequilibrium testing method to check if on average the unaffected parent's PRS is higher than the offspring. And in our study, we found that this was actually significant. The unaffected parent's PRS was on average lower than the offspring's. Next slide. So in conclusion, our study found that RHD has a strong genetic component in Africans. We identified a genome-wide significant genetic region of interest in Africans in chromosome 11, specifically in Black Africans. We replicated previous studies from Ishania's chromosome 11 region in both our Black Africans and our mixed ancestry Africans. We also saw an above average elevated PRS in our probands compared to their parents, and we also identified plausible candidate genes and pathways involved in RHD susceptibility. Thank you for your time. We would also highlight that we have a publication with this data if you want to see further details in JAMA Cardiology. Of note, we also had an editor's note, so it indicates these are very important findings. Thank you.
So looking beyond what we've established right now, there's still a little bit of work that we would like to complete. Tafadzwa is still carrying on with some of the bioinformatics, and she's got enough work to keep her postdoc busy for a while. We also were able to complete a proteomic study in about almost 500 cases and controls, where we tried to see if there were any significant associations between cases and controls. And we've submitted the paper for publication, so you've got to watch this space. And then, as Tafadzwa mentioned, there were studies done in Oceania and other places, Bangladesh, and we are in the process of doing setting up a consortium to conduct a meta-analysis across all of these individual studies. There are possibilities for future functional experiments. And really, in the spirit of Bongani, we are wanting to exploit the partnerships that were created. We have another grant that we've submitted, trying to leverage some of these partnerships as we continue to look at ways of taking the work forward in rheumatic heart disease and certainly in other cardiovascular diseases.
Finally, we would want to acknowledge everyone who participated significantly in this study. We don't want to forget the participation of Professor Bongani Mayosi as he spearheaded the ideology and all of the groups and the people he recruited and included in the study. For example, Dr. Pere, Professor Bernard Kivney, Mike Chong, and Sister V, and all of the different people across Africa and across the world who have helped us to make this study happen. We'd really like to thank the 8 African participating sites: Kenya, Mozambique, Namibia, Nigeria, South Africa, Sudan, Uganda, and Zambia, which formed this RHDGN network. And we'd love to thank all of our study participants as they helped us make the study possible. Finally, we'd like to thank our funders, the Wellcome Trust, UCT, PHRI, and McMaster for all of their support and funding and fellowships. Thank you.
Thank you, everybody.
Okay, so thank you very much for those great talks and the amazing results. We have a couple of questions. We're all running out of time there. So Segun Fatima says, great talk. Do you think a cutoff of 5 × 10⁻⁸ is appropriate for GWAS in the Black African population?
Yes, hi. Thank you for that question. So, so it depends actually. So there are a few factors that cause these cutoffs, the genome-wide significant cutoff. It's mostly linked to Bonferroni correction. So it depends on the number of independent tests you'll be doing. So it depends on your sample size, obviously, if you have enough power, but mainly the independent tests, for example, i.e. the SNPs we have. So 5 × 10⁻⁸ is the common threshold that's used. It is sufficient in some studies, but in other studies, when they're larger, it's not significant. Yeah, thank you.
And there's a question from Michelle Ramsey, just the last question. She may have missed it, but did you have a proposed function for the biology of this chromosome 11 hit in African populations? And are there any signatures for selection in this region?
So this region is actually intergenic. So it's in between a few genes. So these two genes that is mostly in between, it's not clear which one it would be tagging. So what we're planning to do in the future is actually perform further functional analyses. For selection, we haven't fully explored that, but we're planning to actually explore that further, because what we observed is with other studies and our collaborators who we have access to, with other ethnic data sets that are not African or with no African descent, we found that there was actually no chromosome 11 association with increased risk for susceptibility with RHD. So we know for a fact that most probably it's only for Black Africans. Even in our admixed Africans, it's not there. So it seems it's selected in Black Africans. But we're going to do further work on it. So thank you for that question.
I think there's lots of amazing things to explore. There's a further question from Michelle Skelton in the discussion forum, so please go on there, and if there are any additional questions, please have a look for them in the chat and the Q&A and answer them. Thanks again for your talks. So now we're moving on to the last talk of the session. It's an invited speaker, and I apologize in advance for the pronunciation of your name. Our speaker is Carl Schelmoiden, who is he's been epidemiologist, public health practitioner, and many other things, and was a practicing practitioner in medicine in rural and urban areas for 10 years, and then became an academic. So he became a professor at the University of Pretoria and was the founding director of the School of Health Systems and Public Health. And then he became an executive director of CORAD, and he's now ex officio member of CORAD board and president of CORAD USA. And so we're looking forward to your talk about CORAD and fair research partnerships being essential. Thank you, Carl.
Thank you. I just want to be sure that you can hear me.
Yes.
Excellent, thanks. So thanks very much for the invitation that came to talk a little bit about CORAD and what we could possibly offer to the H3Africa group. I'm not going to go into a long history of the organization and all the topics, but want to hone in on one particular one that I think that we feel is very important to this group as a consortium. So I'll start sharing my screen. I hope that that is visible.
Not yet.
Yeah. Is it? Okay, great. So I think the last time I had to deal directly with genetics was actually a 2002 meeting at the University of Pretoria, which to my kind of great surprise and dismay, the rectorate had decided to merge the Department of Human Genetics with general genetics on so-called main campus. So out of the medical school, no more genetics. And we felt it was very short-sighted and not appropriate given the importance that it's taking in the world. And I think the H3Africa Consortium and the work that's being done just shows how kind of limited that decision was at the time. So I'm actually glad to be back in a way, and I certainly hope that I can contribute in a slightly different way. CORED, the Council on Health Research for Development, might be known to some of you. It started with a commission in 1987 that worked for 3 years and came out with this report, which is still pretty relevant to most research and development in low and middle income countries. So the report was called Health Research, the Essential Link to Equity in Development. And it came out with one huge finding. The one overall finding was what was called the 1090 gap, which is, I think, well known, but just to be sure, it indicates that perhaps only 10% of the world's resources was spent on diseases that cause 90% of morbidity and mortality. So there was a huge skewness in distribution of research resources and research capacity around the world to deal with the problems that affected most people. Out of that were 4 major recommendations given. One that focused on a strategy of essential national health research, which has also been applied quite extensively in many countries in Africa, including in South Africa, that focuses and emphasizes the fact that even if you have low health care budgets, spending between 2 and 5% of your budget on operational research, on better targeting, if you want, on priority diseases in the country will help to improve the impact of even marginal budgets. So essential national health research became the core work of what CORED has been doing since 1990. A second major recommendation was to increase global funding for health research substantially. And that has happened in many ways, kind of unrelated to the owners of this report. It is bilateral funding, it is multilateral funding, it's philanthropic funding, there's quite a bit. And again, H3Africa is a great example of how that operates. The fourth, and I'll come back to the third, but the fourth was a global platform to create a place where on regular intervals people could meet to see if that 1090 gap was actually getting closed. And that forum became the Global Forum for Health Research, which demised about 8 years ago due to lack of funding. We've taken over some of that in CORED as an organization, but funding for large meetings is a problem, particularly because we didn't have the mobile capability that, of course, COVID-19 has brought now to bear across the world. The third thing that was a strong recommendation is that the creation of research partnerships to help improve the capability of low and middle income countries to do research. And that is what I want to hone in on. So in 1990, when this report was published, it was one of the recommendations, and partnerships happened by chance or by choice or by personal connections. But increasingly, as you can see from this article out of Nature, I think it was in 2015, there was a whole series on partnerships and research networks, how they are key to developing the capabilities in low and middle income countries. And of course, not just there. Research partnerships are key to any research enterprise, also between high income countries. But our focus is going to be on this and say, how can they help? How can we help? And perhaps work with H3Africa on this to improve the quality and the ability of partnerships to leave. As Dr. Engel was mentioning just in his talk, say they were all over Africa in 8 sources and they want to leave their collaborating centers better off. But then what? Is there a metric? Is there a system? Is there a way in which we can quantify and qualify partnerships in research collaborations? And until now, we found there was none. And so we've been trying to set one up. Many of you who are interested in the topic, you would find that if I go back for 2000, the same articles come back. This one of the first one out of an EDCTP group of grants to look at how research partnerships can help. in the Lancet, kind of key people out of Wellcome Trust, out of people still related, I think, with particularly the Kenyan collaboration of the Wellcome Trust. People out of Uganda, Switzerland put this classical paper now across. In 2014, we were surprised to find that Nature found it necessary to still publish an article with this heading, where it said that partnering with local scientists should be mandatory. So this is now only 7 years ago, when that was still, put up as a key message. But no surprise, I mean, to you will be that as of April this year, the same Nature then published this letter, an open letter to international funders of science and development in Africa, following an award of a 30 million grant to a consortium of American institutions to basically research in Africa for Africa without any African significant participation. And so the issue is topical. Although we talk about it, it keeps coming up. I've been following this now, as you can see, over 21 years, and every time there are other articles, there are recommendations, but then what? And that is what I want to try and talk to you about. So for us, thinking about a new normal in science collaboration should be the acknowledgement and the explicit construction of two parts of that outcome. The one is new knowledge. That's obvious. That is what science is about. That is well developed in terms of metrics, in terms of grants, in terms of career advancement, of how you get up in life. These are the key things that work. That has been tried and tested, and that is good. What is less likely tested is more capable research institutions and systems, particularly in low middle income countries. So we all talk to that. But there is very little in terms of metrics. There's very little in terms of systems. There's very little in terms of guidelines that are being adhered to generically. And so the Research Fairness Initiative that you see there as a last point has been our effort to start operationalizing this second role. So besides the number one, the publications, the excellence of the research, the ethical nature, the contribution and the patents that individual knowledge generations and publication does, there needs to be more. As you all know, it is capable research institutions that generate capable researchers who produce the best research. That is why people go to the top institutions in the world. And so there needs to be this second, okay, we've dealt with the one, how are we going to deal with the other? So over the years, you see that there are lots of guidelines and practices. Often in the first one is for France in French. There are in Canada, the Swiss have their 11 principles and 7 questions. The OECD sets up guidelines. There is a latest global code of conduct for research in resource poor settings. Here's another French guideline for agricultural collaborations. And there are so many, but it's unclear that there is any global standard or there's any global way to adhere to those. So many people have their own internal wish to do better, but they are rarely compared. So the Research Fairness Initiative that we then developed over a long time, including with support, if you want, was a meeting at the Wellcome Trust, but also meetings in Cape Town, meetings in many places in the world to try and understand, is that basically there need to be 2 big components to that. If you as an institution, not you as a researcher, but your institution produces an RFI report, then basically that report asks the institution about questions about its policies and its practices in collaborations. If you go to our website, you'll find the details of those questions, but they are very simple, and research institutions should have those available. For example, a question we find that most institutions actually do not have available, I've never thought about, is one question, is that do you do an assessment of whether your research does any harm on, for example, the health system? And you would be surprised to say, well, how can my research do any harm? Well, the case study that we present is, for example, a clinical trial that would take 3 years to be done in Malawi, and that absorbed 24 fully qualified nurses as trial coordinators in the various centers of the trial. And at that time, about 2008, those 24 nurses represented 50% of the College of Nursing of Malawi output per year. So one trial taking out half of an annual supply of nurses to Malawi, clearly that can do harm to a system. And so the question is, shouldn't you, before you set up big programs, maybe not an individual student project, but big programs, can we actually do any harm? Are we creating opportunity cost problem by hiring staff in one place, they can't do something else? Are worse to ask. And one of the institutions that mostly commented on this was WHO TDR. That was one of the first institutions to complete the report. And they made it very clear that this was the first time that they actually thought about it. And they've since then included that in their reviews, if you want, of large research projects. So there is institutional learning. Once you answer those questions, you can see what is working and what should be improved in your institution over time. If then we get multiple reports from multiple institutions and multiple sectors in different continents, we can start comparing. We can create an evidence base. We can say what works. We can do aggregate analysis. We can perhaps create new benchmarks and standards, for example, on questions like, does your institution have a policy on promoting the role of women in science? Well, if you don't have one, maybe your response to this should be, we are going to develop one in the next 2 years. And so we can then start slowly and systematically build a way to improve the capability and the way institutions collaborate. So when I looked at the H3Africa website, and so my acknowledgement is that is all I really know about this project, is what is printed on the website. So there may be much more to this that is not there. But I find that there is a really good definition of purpose, vision, outcome, participants. So this sentence of the H3Africa aims to facilitate a contemporary research approach to the study of genomics and environmental determinants of common diseases with the goal of improving the health of African populations is pretty clear. But then I ran at a loss. I couldn't find. So what does that mean of the participating institutions? Is there a commitment to having capacity building? If there is, what capacity building? How is it being measured? Is there any way in your presentations or your annual meetings that you actually specify this section and say, okay, what have we done? What have we achieved? What is sustainable? Where is sustainable financing coming in? And so all those parts are missing. And maybe that is one of the contributions that the RFI could make, or that CORED could make to the H3Africa partner institutions of all sides. So the RFI report is not simply for high-income country institutions. That is often what low-income country institutions says. Well, they say, well, fairness means, they have to give more. And it deals with authorship, and it deals with intellectual property right problems, and it deals perhaps with equal salaries for researchers. But that's not the case, because the low-income country institution also needs to state, for example, how it will participate or on what conditions it will grant, what it would like to see in terms of IP sharing, authorship attribution. It should communicate how it will be transparent in financing and finance systems. So it applies to both. It also applies to donors. It applies to government departments that deal with the science and academic sectors and the research sector, so that they can say how, as a country, we would like to make sure that our institutions are being partnered from outside. So the RFI is really a very versatile tool, usable at the key areas at all actors in the institution. It is pretty simple, really, and it's becoming even more simple as we have now had a grant over the last year and a half to make it virtually totally online. So if you have the information available to answer those questions that are kind of listed here in generically, then basically you could produce your own report like in a day. You can enter the information, and the software actually allows you to produce a standard report that, of course, you can then take and make more beautiful. But the basic report can be produced pretty quickly. So the institutional reporting operates at 3 levels. It acknowledges that fair opportunity starts even before the research starts. So who decided on H3Africa? Who decided what the questions were? Who decided that it was a priority for Africa? Was it done jointly? Was it done by the high income country? Was it done elsewhere by a funder? We don't know, but it starts early, and there are a series of indicators that show fairness to all partners concerned before the research even starts. Clearly, the FAIR process deals with the research during research, and you all know that that is a key problem. You know, all the areas where how we collaborate. Another question, for example, that is often not or answered by, oh, we haven't never thought about it, is, but how do you resolve conflicts? Is there a conflict resolution mechanism specifically agreed between the partners? And often that is useful for long-term programs like, for example, H3Africa. And the last, the fair sharing of benefits, costs, and outcomes is what happens after research is over. And that is my particular interest, if you want, in saying, well, by and large, the global health paradigm is very often that a problem is being recognized, let's say in Africa, research is being done and pushed and funded and expertised out of high income countries with or without participation, as you saw in the Nature article, or at least with or with adequate participation of African researchers. But then once it's over, the products and the production, the scientific enterprise, the startups, they happen to be in Seattle around the Gates Foundation, or in London around the Wellcome Trust or the London institutions, or in New York or Washington. They're not going to come to Ghana or Accra or wherever. And so how can we, in this third process, make sure that in the way we engage partners, we can get a better deal also of what happens after the research is being done? So basically, we split those 3 domains into 5 topics, and each has an indicator. And you ask very simply, what are you doing now? If you have any clear written policies, please share them. Or if you have examples, please share them on video so that we can share them with others. And then thirdly, what do you want to improve over the next 2 years? And so the RFI is a mechanism to help you focus on how to improve. It is not meant as a report card. So it is meant as a progressive quality improvement tool. Perhaps the last slide to show you that it's slowly being picked up across high-income countries, low-income countries, international organizations. We are very happy also because the invitation to speak came out of the University of Cape Town. So I don't know if you were aware of it, but the University of Cape Town has completed its internal review. So the report is ready, but it's now with the executive to be finalized and approved for publication. And it is important to mention that it was the new rector, the first woman, the first African woman as the rector of the University of Cape Town, who felt that for a new mandate of this university, the RFI provided a key instrument for UCT to become a fair partner also in Africa, not just as a receiver, but also as a kind of a lead organization in many cases. And you see that Caprisa is starting up, a major AIDS research center, the biggest AIDS research center, I think, in Africa. Epicenter Paris basically is the clinical trial arm of Doctors Without Borders. Areas like Palestine, who are really at the bottom of research development, are seeing it as a way up, etc. So in a way, there are many that are picking it up. And we would hope to be able to work with some of you to see if this can be something that would help the institution to become, if you want, a better beneficiary and potentially a better contributory to partnership building and research, institutional research capability building in Africa. Let me leave it there.
Thank you very much, Carl. So that was a really interesting talk. And we have a couple of comments from Michelle Skelton. She said the RFI sounds very useful. It would be great to explore and apply this. And then from Suhail, that she really enjoyed your talk, very insightful, food for thought for low-income countries, especially with respect to training and capacity building. This is something we need to look into and explore. So how practically do you think, what are the practical next steps for H3Africa in terms of exploring this?
I think there are two ways in which you could do that. One is if you feel that your institution is already ripe to do so, you can go individually, of course, to the top of your institution, bring this across. If you want, set up a webinar. The director of our RFI program is actually based in South Africa, so she can connect through electronic means and have a conversation to help the institution get across. For details with H3Africa, because there are many different partners involved in that program, I think it might be good to have a meeting specifically on this and say, so how can we tackle this? How can we make it come alive and not fear that it is more administration or more work, which we particularly don't want to do, and say that actually, if you think about the questions that you can see that you can download from the website on the RFI, is they're all questions that a responsible research institution should be able to answer. You should be able to answer that you have a policy on women in science, or you should be even, to give you an idea, and I speak a bit about, I think with permission, but for example, TDR had the WHO TDR, they had their first round on this, and they were not aware, and they were kind of worried that when we asked about this question about do you have a strategy for role of women in science, but when they started digging through their scientific advisory board, there was actually a recommendation that 50% of all grants should go to women PIs, and when they then started counting their own grant mechanisms, they found that it was in the order of 42%, and so that's a pretty high percentage. It's pretty good to share that kind of information, and of course you can get better, but that is how it could work individually. That is how it potentially could work in the H3Africa. And for example, some key questions that we asked, we could send around in the H3Africa and say, well, are you satisfied with authorship divisions? Are you satisfied with how various of those questions are being dealt with in the partnership?
Those great suggestions, and please look out on the Q&A for some live chats. We have a break coming up now, and we're a little bit over time, but I think many of the PRs will be really interested in engaging with you further. So please keep an eye out on the Q&A and answer those questions. So I'd just like to thank all the speakers from this session, and we're having a break now, and there is an exercise session organized if you'd like to join that, and please rejoin us at 3 p.m. CAT or whatever time zone, whatever time it is in your time zone, and that'll be for the fellows speed presentations. And we enjoy your break, and we look forward to seeing you then.
Thank you.